The creation of a new human life is an incredibly detailed process. It begins with a single fertilized cell. This cell contains a complete genetic blueprint. This blueprint guides the development of every system in the body. Among the most complex of these is the reproductive system. Its formation is a testament to biological precision. It involves a sequence of timed events. Each step must occur correctly for the system to function later in life. This journey transforms a simple embryo into a being capable of creating the next generation. It is a story of cellular signals, hormonal guidance, and intricate construction. Have you ever wondered how the female reproductive system actually forms? It does not simply appear fully formed. Instead, it develops through a remarkable and delicate process. This process begins very early in prenatal life. It continues with significant changes during puberty. The initial stages are common to both male and female embryos. A critical turning point then directs development down one of two paths. Understanding this journey offers deep insight into human biology. It also helps us appreciate the complexities that ensure the continuation of our species. The story is one of potential, differentiation, and maturation. This essay will explore the amazing journey of female reproductive system development. We will start at the very beginning, inside the womb. We will see how an embryo, initially without clear sexual characteristics, is guided to become female. We will look at the key structures like the Müllerian ducts and the hormones that act as messengers. The formation of the ovaries, uterus, and other essential organs will be explained step by step. We will also touch upon what happens when this intricate process deviates from its typical path. It is a journey that showcases the elegance and complexity of nature's design. From the first few weeks of gestation to the transformative years of adolescence, the female reproductive system undergoes a profound evolution. This development is not a single event, but a long and carefully orchestrated symphony of biological processes. It involves the migration of cells, the fusion of tissues, and the powerful influence of hormones. By breaking down this process into its key stages, we can gain a clearer understanding of how this vital system is built. This knowledge is fundamental not only for students of biology, but for anyone curious about the human body. It highlights the precision required for health and fertility throughout a woman's life. In the early weeks of embryonic development, the potential for both male and female structures exists. Around the 5th week of gestation, every embryo is equipped with 2 sets of primitive ducts. These are the Wolffian ducts and the Müllerian ducts. At this stage, the embryo is considered sexually indifferent. It has not yet committed to a male or female developmental pathway. This shared beginning is a crucial aspect of sexual differentiation. The future of the reproductive system hinges on which of these 2 duct systems is instructed to develop, and which is told to disappear. The decision is made by genetic and hormonal signals. The key to this decision lies in the sex chromosomes. An embryo with XY chromosomes is genetically male. An embryo with XX chromosomes is genetically female. The Y chromosome carries a critical gene called the SRY gene, or sex-determining region Y. In a male embryo, the SRY gene activates around the 6th week. It triggers the development of the primitive gonads into testes. The newly formed testes then begin to produce 2 important hormones. These hormones are testosterone and anti-Müllerian hormone, or AMH. These 2 substances are the primary directors of male reproductive development from this point forward. In a genetically female or XX embryo, there is no Y chromosome. Consequently, there is no SRY gene. Without the SRY gene signal, the primitive gonads do not develop into testes. Instead, they will follow a different path to become ovaries, though this happens a bit later. More importantly for this stage, the absence of testes means there is no production of testosterone or AMH. This absence of male hormones is the single most important factor that allows the female reproductive system to develop. The female pathway is often considered the default pathway because it proceeds without these specific male hormonal signals. Therefore, the fate of the 2 duct systems is sealed. In the male embryo, testosterone stimulates the Wolffian ducts to develop into the internal male reproductive structures like the vas deferens and seminal vesicles. At the same time, AMH actively causes the Müllerian ducts to degenerate and disappear. In the female embryo, the opposite happens. Without testosterone, the Wolffian ducts fail to receive the signal they need to survive and grow. They gradually wither away. In the absence of AMH, the Müllerian ducts are free to continue their development, laying the foundation for the internal female organs. Once the Wolffian ducts begin to regress in the female embryo, the Müllerian ducts take center stage. Between the 6th and 12th weeks of gestation, these paired ducts undergo a remarkable transformation. They are the raw material from which the core of the female reproductive tract will be built. This development is a process of elongation, fusion, and differentiation. It happens in a specific sequence, starting from the top and moving down. The upper parts of the Müllerian ducts remain separate. They will eventually develop into the two fallopian tubes, the delicate channels that will one day capture eggs from the ovaries. Further down, the two Müllerian ducts move toward the midline of the embryo's body. Here, they begin to fuse together. This fusion is a critical event in the formation of a single unified uterus. The upper fused portion becomes the main body of the uterus, while the lower, narrower fused portion develops into the cervix. The wall that initially separates the two ducts in the middle must completely break down and be reabsorbed. If this process of fusion or reabsorption is incomplete, it can lead to structural anomalies of the uterus, such as a bicornuate or heart-shaped uterus or a septate uterus. The development continues downward from the newly formed uterus and cervix. The very bottom tip of the fused Müllerian ducts connects with another structure called the urogenital sinus. This connection point initiates the formation of the upper part of the vagina. The lower part of the vagina develops from the urogenital sinus itself. A solid plate of tissue known as the vaginal plate forms and then canalizes, meaning it hollows out to create the vaginal canal. This process is typically completed by the 20th week of pregnancy. A small membrane, the hymen, often remains where the vagina meets the external genital area. By the end of this period, the internal architecture of the female reproductive system is largely in place. The fallopian tubes are positioned to connect with the future ovaries. The pear-shaped uterus is formed and ready for its future role in pregnancy. The cervix provides a channel between the uterus and the vagina. The vagina itself is formed as a muscular tube connecting the cervix to the outside of the body. All of this intricate construction occurs silently within the developing fetus, guided by the absence of male hormones and a precise genetic program that directs the Müllerian ducts on their developmental journey. While the Müllerian ducts are busy forming the internal organs, a parallel process is happening with the gonads. In the female embryo, the undifferentiated gonadal tissue is destined to become the ovaries. This process begins a little later than testis development, typically around the 8th week. The initial step is the organization of the gonadal ridge into a primitive ovary. This involves the development of a cortex on the outside and a medulla on the inside. Unlike the testes, the ovaries do not produce significant hormones at this early stage. Their primary role is to house the future egg cells. The egg cells themselves have a fascinating origin story. They do not arise from the ovarian tissue itself. Instead, they begin as primordial germ cells in a completely different part of the embryo, near the yolk sac. Around the 3rd week of gestation, these primordial germ cells begin a long migration. They travel through the embryo's developing gut to reach the gonadal ridges. This journey is essential for ensuring the gonads become functional. Once these cells arrive at their destination around the 6th week, they populate the developing ovary and are now referred to as oogonia. Once inside the ovary, the oogonia begin to multiply rapidly through cell division. Their numbers increase dramatically, reaching a peak of several million by about the 5th month of gestation. This is the maximum number of potential egg cells a female will ever have. Following this period of proliferation, the oogonia then enter the first stage of a different kind of cell division, called meiosis. As they enter this stage, they are called primary oocytes. Each primary oocyte becomes surrounded by a single layer of supportive cells, forming a structure known as a primordial follicle. This process protects the oocyte and holds it in a state of suspended animation. This process of oocyte development and follicle formation is unique. No new oocytes are formed after birth. The vast number created during fetal life is all a female will ever possess. From the peak of several million, the number of oocytes begins to decline even before birth. Many undergo a natural process of degeneration. At birth, a baby girl has approximately 1 to 2 million primordial follicles in her ovaries. This number will continue to decrease throughout her childhood. The oocytes within these follicles will remain arrested in their first meiotic division for years, waiting for the hormonal signals of puberty to awaken them. The development of the external genitalia also follows a principle of common origin. In the early embryo, before the 9th week, the external structures are identical for both sexes. This undifferentiated stage includes a small bud called the genital tubercle, two folds of skin called the urethral folds, and another pair of swellings called the labioscrotal swellings. The appearance is neutral, with the potential to become either male or female genitalia. The final form these structures take is determined entirely by the presence or absence of powerful androgen hormones, specifically dihydrotestosterone, a potent form of testosterone. In a male fetus, the testes produce testosterone, which is converted to dihydrotestosterone in the target tissues. Dihydrotestosterone is a powerful masculinizing hormone. It causes the genital tubercle to elongate significantly and form the penis. The urethral folds are stimulated to fuse along the midline, creating the shaft of the penis and enclosing the urethra within it. The labioscrotal swellings also fuse together to form the scrotum, which will later house the testes. This entire process creates the distinct external anatomy of a male. It is an active process driven by strong hormonal signals. In a female fetus, the absence of testes means there is no significant source of testosterone or dihydrotestosterone. Without this potent hormonal signal, the external structures follow the female developmental pathway. The genital tubercle undergoes very little growth and becomes the clitoris, a much smaller and highly sensitive organ. The urethral folds do not fuse together. Instead, they remain separate and develop into the labia minora, the inner folds of skin surrounding the vaginal opening. The urogenital sinus remains open to the exterior, forming the vestibule where the urethral and vaginal openings are located. The outermost structures, the labioscrotal swellings, also follow a different fate in the absence of dihydrotestosterone. Instead of fusing like the scrotum, they remain separate and enlarge to become the labia majora. These are the larger outer folds of skin that protect the other external genital structures. This entire process of female external differentiation is largely complete by the 14th week of gestation. It serves as a clear example of how the absence of a specific hormonal signal is just as important as the presence of one. The female form emerges naturally when the masculinizing instructions are not delivered. For more than a decade after birth, the beautifully constructed female reproductive system remains largely dormant. The ovaries contain their lifetime supply of follicles, but they are quiet. The uterus is small and inactive. This period of calm is known as childhood. The hormonal signals needed to awaken the system are kept at very low levels. The brain, specifically a region called the hypothalamus, is the ultimate controller of this process. During childhood, the hypothalamus is highly sensitive to the small amounts of estrogen produced by the ovaries, keeping the system in check through a negative feedback loop. Puberty begins when the hypothalamus becomes less sensitive to this feedback. This change allows it to start releasing a key hormone called gonadotropin-releasing hormone, or GnRH, in rhythmic pulses. The release of GnRH is like turning the ignition key for the reproductive system. GnRH travels a short distance to the pituitary gland, a small but powerful gland at the base of the brain. In response to the GnRH pulses, the pituitary gland begins to produce and release two other critical hormones into the bloodstream: follicle-stimulating hormone and luteinizing hormone. Follicle-stimulating hormone and luteinizing hormone travel through the blood to their target destination, the ovaries. Their arrival signals the end of the long dormant period. Follicle-stimulating hormone, as its name suggests, stimulates a group of the primordial follicles to begin growing and maturing each month. As these follicles grow, their cells produce increasing amounts of the primary female sex hormone, estrogen. Luteinizing hormone also plays a role in follicle development and is responsible for triggering ovulation, the release of a mature egg from the follicle. This new, cyclical production of estrogen by the developing follicles is what drives the physical changes of puberty. This surge in estrogen has widespread effects throughout the body. It causes the breasts to develop, the hips to widen, and body fat to be redistributed in a typical female pattern. Estrogen also stimulates the growth of the uterus, vagina, and fallopian tubes, preparing them for their adult functions. The most visible milestone of this process is menarche, the first menstrual period. It signals that the hormonal cycles have begun and that the reproductive system is maturing. This intricate hormonal cascade, starting in the brain and ending in the ovaries, transforms the body and awakens the reproductive system, making it capable of reproduction.